right, well, great. It's a pleasure to be here. It's a different format than I usually have the opportunity to speak in. So I have a lab at Mass General Hospital, kind of full of physician scientists and basic researchers and postdoctoral scientists. It's more of a classical lab where people are focused on individual projects. And I also have a group at the Broad Institute, which probably you've seen people from the Broad come over and speak here, where we work on a sort of a different big science model and a lot of people working together to try to understand how the genome works and collaborations and bringing in new technology. So it's kind of nice to be at the interface of both those worlds of research. Anyway, it was nice to get this invitation and both to give back a little bit to teachers. I really appreciate all that you do. Some of you are probably having to deal with my son on a regular basis. But also it did cause me to think back. There was, I had this amazing couple teachers that I had, particularly in middle school in Seattle, in sort of a program for kids who were a little overactive. And Mr. Isker was one I remember in particular, who just drove kids in their creative, just had a way of bringing out the creativity in the kids. And I stayed in touch with him every few years afterwards. I haven't talked to him in a while. I probably should. But it was really cool because I just noticed that Broad just hired this real hot new associate member, core member actually, that they brought out of a fabulous lab in Stanford. And we got to talking, and it turned out that we both had Mr. Isker in Seattle, which just blew me away. So this guy made an impact. It's very special. This is a complicated topic I'm going to dive into, but this is what drives my research and drives me is, this simple question. And how does one genome, right? We all have our genome, and yet we have this genome through development gives rise to so many different cell types in our body that are so dramatically different. And I'll talk a lot about that, but it's obvious when one looks at, you look at each other, look in the mirror, what we call them the phenotypes, or the way a cell looks is so distinct. The genome has this information, but how it is manifest in all these different contexts is the subject of my research. And what I want to talk about today, the genome's big. It's the human genome. There's been a lot of excitement because the genome was sequenced about a little over 10 years ago, and we had all these celebrations at the National Human Genome Institute, and found 3 billion bases of letters. But it's hard to interpret. It's kind of like a Russian novel, but you don't speak Russian, and there's no spaces between the words, right? So you can't even tell where a word is and where is the information unit. You can use computational methods to find these genes. So there are, that the DNA contains genes that make RNA and then protein, and so those are what code for your traits, like your eye color, and it's because the gene codes for a protein. It goes through DNA to RNA to protein. You can find those computationally. And with the Human Genome Project, all of a sudden people had a good idea. There's about 20,000 genes in the human genome, which is actually quite a bit less than people had initially thought, and actually about the same as a roundworm. So it kind of makes you scratch your head a little bit about how we are this marvelous that we are, how we could have about the same many genes as a roundworm. But the other striking thing is that if you added up all the genes and you figured out how much of the genome that those genes made up, it's about 1%. So 1/100th of those 3 billion bases code for proteins. And so what about the other 99%? Right? This was kind of a mystery that, so as the Human Genome Project finished the draft, this became a great mystery. How do we understand that? And people thought, well, this is junk DNA or it's dark matter. But really we're learning now that huge amounts of this remaining 99% are incredibly important, and they really explain much of this question about how one genome gives rise to so many cell types. So I just made this point that the protein coding genes are just about 1% of the genome. So I want to talk about how do we find the genome's other working parts, right? There's sequences all throughout the genome that do things, that regulate things of how our genome works in different contexts. I'm going to talk about how we can find those and how we can understand how those work. How do we understand the rest of the genome? And really then a little bit about how these parts interact to give rise to, there's more than 200 different cell types in a person, right? So we're really complicated. And that's probably a big underestimate because in the brain things get so complicated. So one genome, so many cell types, right? Not only within an individual person, do you have the same DNA making these different cell types? You can actually show this in the laboratory, and I'm guessing you've had other lectures from we have some spectacular stem cell biologists in the building here or across the street who can take either ES, embryonic stem cells, right, cultured in dishes, and can, through these sort of complicated additions of growth factors and conditions, can make neurons and all sorts of other tissues. And here again is an example where you have this one cell, and the cell's not changing its genome, but it's dramatically changing its phenotype, right? So how does this happen? This is a complicated slide, but I'm going to now, after this slide, give you a bunch more slides that basically say the same thing, again, just to really clarify this really critical point. I mean, this is sort of a classical, the field of epigenetics sort of was driven by this guy, Conrad Waddington, who in 1949 described Waddington's landscape. And he said, well, you know, they didn't really understand the structure of DNA. Well, they certainly didn't understand the structure of DNA or how the genes were made up. But he came up with the idea that somehow cells during development would roll down these slopes and occupy different energy minima, right? So you can imagine a ball rolling down the hill and making choices. And his picture he drew here is a lot like when we see pictures of development, like this might be a hematopoietic stem cell or an embryonic stem cell, and then you have a hematopoietic stem cell that then differentiates into myeloid cells that make up, that make up some, blood cells, and then the lymphoid cells that make up your immune system, right? You can imagine that these cells are making decisions along the way, like they're rolling down a hill and choosing different paths, right? So what is the basis of this? Well, we have about 20,000 genes. The genes in all your cells are the same genes. You have the same DNA, but in different cell types, you turn different genes on or off. And that's really the basis of epigenetics, is that different cells make different proteins. So you have 20,000 genes, but probably only about a third or a half of them are actually making proteins in any given cell. And at the end of the day, what makes these cells so different is they're making different proteins. So the proteins are the machines that make the cell what it is, and the regulation is just critical to what genes come on and what genes are making RNA and protein and which are not. A lot of what I'll talk about then is how the genomic DNA, just shown here, is organized in the nucleus, and how it's organized in different ways in different cell types to accomplish different gene expression programs. Okay, and I'm going to tell you the same thing again. I think found some very nice pictures. There was this recent report by the ENCODE project. There's a sort of a post-genomic era project called ENCODE that sought to find all these pieces, the working parts in the genome, and it was reported in the New York Times recently. And they actually, I'm not sure if these originally came from the molecular biology of the cell, but I think it was sort of altered in the New York Times article a little bit to try to explain some of these concepts, such as, you have these stem cells early on that have this open potential. Their options are open, and they can form different kinds of cells in the body. For example, a liver cell or a neuron, just one of the most dramatic examples of how different things go. They have the same DNA, but the way that DNA is packaged is very different, right? So this is what I just told you. I'm telling you again because it's really central to this. And here's some pictures to help you understand what we mean here. So the DNA, actually, I'll show you. Let me see if I've got a better picture. I'm going to jump back to this. Let me start by showing you how the DNA is organized in the nucleus. So the DNA is about 2 meters long. So in a single cell, you have about this long stretch of DNA, right? And it's obviously it's not very thick, but it's a very narrow string. But somehow this 2 meters needs to fit into a nucleus, which is the size of a head of a pin. So how is this accomplished? Well, here's the double helix. We'll come back to this a little bit later, but it's wrapped around these spools of histone proteins, and they're called nucleosomes. And so the fundamental building block is the nucleosome. It consists of about 200 bases of DNA wrapped around these nucleosomes. And so the whole genome is wrapped around nucleosomes and around spools. It's a very elegant way, I think, of packaging a very long string. If you had to package up a very long string in a very small space, this might be how you do it. You wrap it around spools, right? And then it turns out that these spools sort of tie together in very intricate combinations and package in, and they form sort of larger structures of chromatin, that then condense and form what we think of as the chromosomes, which we can visualize. So there's this very intricate packaging of the DNA to fit into the chromosomes and then fit the chromosomes in the nucleus. And the point we wanted to make here is that actually this packaging is not solely a packaging. It's actually not only a packaging, but it's also it has regulatory functions. By the way, if I go too fast in any way, stop me and feel free to ask questions. I meant to say that at the beginning of my talk. So here might be the structure of if you had a gene that wasn't used. There's a lot of genes actually that aren't used. For example, you've got all these genes encoding olfactory receptors that help you smell. There's hundreds of these genes, and they're never expressed anywhere except for in this olfactory bulb, responsible for smell. Never expressed in any other cell type in the body. So they're always going to be packaged really tightly around these spools, and the spools come together and they're compact and they're inaccessible, and those genes just never come on in other cell types. They only come on one of them comes on in each olfactory bulb. On the other hand, you might have another gene, such as in blood cells, you might have these genes that are sort of turning on hemoglobin, or other genes important for like a neuron, for a synapse or a receptor, right? Those genes, the DNA has the code and the sequence to make the RNA. Those genes need to be open. So actually, they aren't wrapped in so many nucleosomes. There's some spacing between the nucleosomes, and even the nucleosomes will spread apart and allow RNA polymerase, which is what takes that DNA code and synthesizes RNA off of it, which can then go off to a different part of the cell and make the protein, allows that to access the gene. And so this might be what a gene in a cell that's on would look a little more like this. So it's really a structural problem. It's a real contrast between this DNA structure and this DNA structure. And there are whole sets of enzymes and regulatory proteins whose job it is to either compact like this or open like this. So people, you may have heard about epigenetic modifications or chromatin modifications. It turns out that these are the histone, these spools, they're called nucleosomes or sort of histone. The proteins are histones. And the histones have these tails. And one of the really exciting developments in biology that's incredibly recent, over the last, say, 20 years, people have realized that the histones are, these tails can be chemically modified in all different ways. You can add methyl groups on there, just the CH, and you can add acetyls on there. You can phosphorylate the tails. And I'll show you a little more, but these tails are modified in a lot of different ways. And it turns out that when tails get modified in particular ways, they form binding sites that these proteins will sometimes recognize and dock into. So they'll pull in proteins. It's a good way to nucleate a structure, right? So there are these repressive structures that can be recruited to the nucleosomes and help stabilize this very repressive structure. There's also other kinds of modifications, different kinds of chemical modifications that you might put on a histone that would recruit other things that might open, remodel. There's actually remodelers in the cell that take these DNA and they unwrap it and they move the nucleosome around. And there's other kinds of modifications that will recruit remodelers. So yeah, so again, now just to go back, so here's the DNA, picture of the DNA. It's really the whole genome is pretty much wrapped around these nucleosomes, folded into these chromosomes, and there are these modifications. One kind of modification you're probably familiar with is DNA methylation. So the DNA bases, not only do you have the bases of A, C, G, or T, in certain cases the C can be methylated. So it can be modified directly. In a lot of other cases, you actually modify the histone tails. You can think of them as chemical modifications, or it might be easier just to think of them as tags. The cell puts a tag on some of the nucleosomes at a gene, and that tag lets the cell remember, this is a gene that I need to keep on. Right? And then there's a different kind of tag that the cell will put on a gene that, like for example, those olfactory receptor genes that you just never need in any other cell type. You might put a different kind of tag on those genes so the cell remembers to just keep them off and maintain their inactivity. So part of, I guess I'll get to, I'm going to get to the concept of epigenetics in a minute, but really about maintenance of active state or inactive state really goes to the heart of the concept of epigenetics. I'm going to get to that in a minute. But this is just to show you, give you a little idea of why it would matter. So this is another way of looking at that spool. The DNA is purple here, and these are the histone tails. The histones, the red is the histones. There's these tails that kind of wrap around. The tails are positively charged, and the DNA backbone is negatively charged. So you can imagine this forms a real tight structure. It's not very accessible. It's hard to transcribe. It would be hard to make RNA from this. But there are enzymes called histone acetylases, which come in and they acetylate these tails, which neutralizes the charge, which would open up the tails, loosen the interaction, and now all of a sudden the DNA could be pulled off of here as needed to make RNA. So now here's a good structure for a gene to come on. So these are activators. And then there are also histone deacetylases, and these are an enzyme that removes that acetyl tag. And those are repressors. Okay? I also told you that there are enzymes that recognize those modified tags. So this is one that would bind to an acetyl tag, and it will remodel a gene, could turn it on. And then I love this picture here, because this is actually real data here. This is from one of our colleagues at Harvard, Bob Kingston, who actually took a picture, electron microscopy picture of those nucleosomes. So this is DNA, and these little balls here are actual electron micrograph pictures of those nucleosomes, where the spools, right? So you can actually see what this looks like. Here's DNA, and here's the spools. And there are these repressive proteins that recognize a particular tag that turns things off. And this is what happens if you add them to the soup. All of a sudden, everything balls up and compacts. So I think this is a nice sort of visual picture of how in a cell, by these chemical reactions, you could either take DNA and you could open it up and let it be transcribed, or you could compact it in a tight little ball that would make it very inaccessible and difficult to use to transcribe that code into a protein. All right, epigenetics is tough. We've gotten a lot of press, some of which is just, kind of a little bit over the top. And I'd be happy to discuss it if people have questions about epigenetics and, mothers' diets or grandmothers' diets or, flame retardants or other things like that. I mean, the truth is we don't really understand a lot. We're just starting to hit the cusp, and people have talked a lot. But I want to try to step back and just tell you, in general, you start with some safer terms. The field of epigenetics really seeks to understand non-genetic changes, so those changes on top of the DNA, not changes to the DNA sequence, but changes, for example, to how the DNA is organized or wrapped that influence which genes are on or off, and thus change the way a cell's phenotype, how a cell behaves. So these changes may include those modifications like the DNA methylation or the modifications of the histones and the nucleosomes I just showed you, but they can also encompass many other entities. And I'm going to talk, we should broaden it. And the concept are, how can you keep a cell in a stable state, in a stable behavior that's remembered throughout the lifetime of an organism or something like that? How does your muscle cell remember to stay a muscle cell throughout your whole lifetime, and it doesn't turn into a neuron by accident or something like that, right? That's kind of what epigenetics is about. Clearly, these changes can remember it across a lifetime. We know that. So in your body, your liver remembers to be a liver. You got these stem cells that remember what they should be, and they turn into other cells. There's a lot of controversy about this one, though. They may rarely be inherited across generations. So I'm a big skeptic. Usually, I really like to see something. You got to hit me in the head before I'm going to believe it. In plants, it's clear as day that plants there can be transgenerational epigenetic inheritance. Plants will actually seem to learn when, if a seed's dispersed and it goes in a place where there's an early, where the spring comes early, it'll remember that and it'll help it flower earlier. If the seed lands in a place where the spring comes late, like I guess here or maybe in New Hampshire, it'll remember that and it'll tend to flower a little later. And actually that seems like a perfect mechanism for epigenetics, right? You don't want to change the DNA sequence every time a seed happens to fly into a different, New Hampshire and then down into Rhode Island. It's a perfect example of epigenetics. Why not get a trait that you can modify to this better fitness for this environment? But of course, if it's in plants, now people think, well, this may be in humans and in mice. And there's been a lot of research in the field now to try to understand whether, whether some of these can be inherited across generations. I'd be happy to talk about it in the question and answers a little bit later. But at this point, I think, it's still pretty unclear whether this happens and whether any of these things I'm talking about actually contribute to, something your grandmother ate, having some effect on you getting diabetes. So one of the classic examples of epigenetics, here is a change on top of the DNA sequence that doesn't change the DNA sequence but has a big effect, this is a kind of a cool one. It's the Calico cat. It has to do with X chromosome inactivation. So males have, in addition to the autosomes of the X chromosomes, males have an X and a Y chromosome. And they're both open, and they're both sort of allowed to make genes that needed to make genes. And but here's the interesting thing. It turns out you can't really have two copies of an X chromosome. Females do have two copies, but you can't make genes off both copies. That would be, the cell doesn't tolerate that. The cell wants to have effectively have a single X chromosome copy. So how does the female cell, the female nucleus, handle that? Well, what it does is it epigenetically inactivates one of the X chromosomes. So in fact, you have two X chromosomes, but one is completely compacted. Remember those structures I showed you of the really shut down nucleosomes and chromatin? It turns out that in all female cells, one of the X chromosome is inactivated. So it's like it's not there. The DNA is still there, but it's like it's not there. Not making any protein, not having an effect on the cell. Now that inactivation sort of happens during development and can happen in sort of a clonal patchy way. So you can imagine an embryo forming different sections of the embryo. Some sections have inactivated the first X chromosome and some sections have inactivated the other X chromosome. It's random which one gets inactivated. But once it gets inactivated in development, all the progeny of these cells will keep that same one inactive.
What happens in the case of an X trisomy?
Is there an X? There's XXY and X.
Is there an XXX?
There is. You see two bar lines.
Yeah. Oh, okay. So that answers the question.
The collective wisdom in a room is just incredible. We could give this lecture if I wasn't here, you could give this lecture. Thank you. So you have to inactivate both. That would make perfect sense because, right, you can only tolerate one X chromosome. So the Calico cat is really cool, right? So it turns out there's a coat color on the X chromosome, and if the Calico cats actually have different the coat colors, one coat color is on one X and one coat color is on the other, and the X chromosome gets inactivated early, and so as that cell divides and forms more and more of the skin, the whole patches that came from that same stem cell have inactivated that same X chromosome. So they're all going to be the same color. And then there'll be other patches that have inactivated in other ways, inactivated the other X chromosome. So you get this sort of patchy pattern.
So the idea is that the maternal and paternal X chromosome, different ones are turned off at different points in the embryo, and so parts of female body are expressing their maternal X, and other parts are expressing their.
That's right, the maternally and paternally. But here's one key: is the inactivation is kind of random. There are genes that are. I could talk to you about genes that you get from your father and genes that you get from your mother. This is not the case of that. In this case, you're absolutely right. You inactivate your maternal or your paternal, but the inactivation is random. You're not just inactivating the one you got from your mother or just inactivating the one you got from your father. You're sort of randomly, in different patches, inactivating one or the other. But you got it exactly right.
Does this happen in all the rest of the autosomes too? Then we can't have a double expression?
Only on the X.
Only on the X.
Yes. There's something else that goes on the other autosomes I could talk about, but
Okay, I don't mean to take you off topic. I'm sorry. Thank you.
no problem. Question about?
You were saying that if you had 3 copies, 2 would shut off, right?
Well, we've learned that from the audience, but
But what happens in like a Turner-syndrome case where you just have one X? And if you only have one, still not much.
well, then you wouldn't need to inactivate. Yeah, you wouldn't inactivate. a good question. Yeah.
So it's the same in humans, right? So what does it do to color blindness in women? Shouldn't there be women which are colorblind on one eye, or?
Interesting question. I hesitate to answer that, but I would think that often when there are male traits, that the fact that there's random inactivation overcomes that problem. But I think that would explain it. You have both copies of the X, they're randomly inactivated. That said, there are cases where you skew in one direction, so females can get some of these traits that are normally male traits, and those are because you have a skewing that you more often are inactivating the one you inherited from your mother or something like that.
So you're saying when they shut one of them off, it's more or less 50-50 throughout the body?
It's random which one they shut off, but it happens early in development, so it can be patchy.
Okay, but for instance, his question was saying maternal and paternal. You're expressing like 50% maternally throughout your body and 50%.
That's the idea, but there can be skewing of that ratio.
Do you know if this has been studied in identical twins?
If X inactivation, so
I think the activation happens well after the identical twin split.
right, it
Well after.
does.
Really early.
It does. I guess so. So probably that's right. So I think that it does happen after the twins are split. I think there's an interesting question, and that is, are there some epigenetic features that are shared by identical twins that we could talk about? In this particular case, the inactivation happens after the twins are split, which is a fair point. Good. Great questions. Why do people care about epigenetics? People care about epigenetics a lot right now, and there's a lot of reasons for that. One is cancer. Cancer is a really big deal right now, and there's the cancer genome projects, and people are realizing that there are loads of mutations, and when you learn about those mutations, you can begin to target drugs against those mutations. So cancer is a genetic disease, but it's also an epigenetic disease. It's an epigenetic disease because the genes that are turned on or off in cancer are aberrant. You lose those careful patterns of turning just the right genes on and just the right genes off, and at its root, cancer is a gene of aberrant gene expression. The epigenetics is aberrant. There's also inappropriate patterns of DNA methylation. And something that's very recent is it's turning out that, I showed you those enzymes, those deacetylases and acetylases that put the tags on. Really big news over the last 2 or 3 years has been that those enzymes get mutated in cancers. They get inactivated by genetic changes. They just shut off those enzymes, they kill them. And that is a big part of cancer as well. So cancer, you have to change into this other cell type. You have to do a lot to change the epigenome, to change how the DNA is organized into chromatin. It also turns out that neuropsychiatric and metabolic and developmental disorders, they have this sort of environmental component, but they also have a lot of evidence that there's epigenetic components that are underlying a lot of the things that we're dealing with today in complex diseases. I'm going to come back to this a little bit. And finally, there's this really interesting feature that as I mentioned before, is quite speculative at this point, but there are these long-term health consequences of early environmental exposures. And a big question in the field is, are these mediated through the epigenome, as we've talked about? So part of this lecture is genome, and you can't really study the epigenome or how the genome is organized unless you understand a little bit about genomics. And I don't know if you guys have learned this, but have we gone over some of the DNA?
We're AP teachers, we're okay. We're advanced biology teachers, we're okay.
Good. So have you learned about DNA sequencing technologies? Okay, so I don't need to go into all of this. An extraordinary thing happened a couple years ago. And that is, in terms of, if you look at how much it costs to sequence a genome, the Human Genome Project cost about $100 million to do one draft human genome. And because of that, it really drove costs down. So pretty soon you could do, 5 years later, you could do a human genome for $10 or $20 million. But look what happened here. In about 2008, all of a sudden the price of a genome just dropped at a striking rate. So what's the basis of this? What happened in 2007? And what happened was the development of sort of next generation sequencing instruments. And this is a big part of what I'm going to tell you about and how we study chromatin and how we study the epigenome. So I thought I would just at least mention this quickly. It's fundamentally different from Sanger sequencing, which is done, won a Nobel Prize. It was fabulous, but it requires these gels. And you guys have probably done this at some point. You run gels and you look at a base and you find the terminator and you do 1, 1, 1, 1. The next generation sequencing actually does this in a different way, and that is they do it by sequence by synthesis. So you would attach lots of pieces, small pieces of DNA onto a slide, and they're spatially segregated. And you have a camera, a very high resolution camera, that watches those spots of DNA. And what you then do is you add each of the A, C's, G's, or T's, and you take that strand, you elongate the other strand, you build that other strand. And each time a base gets added, it emits a fluorescent piece of light. And there are 4 bases, and there are 4 different colors. So what you do is you add each base, you add one base, you actually add all 4 bases at a time, and you take a quick snapshot, and it just told you what base 1 is. If it's green, it's an A. If it's yellow, it's a T or something like that, right? And you just do this in recurrent cycles again and again and again. So you take one picture, there's a terminator on it, you remove the terminator, you add the next base, and you watch the picture, you see what color it is, you remove the terminator, you add the next base. And I mean, technologically, this has been so transformative to science in the last 5 years, it's hard to overstate. The chemistry is quite remarkable in terms of being able to add these different dyes and capture this. But I think what might be actually at the end of the day driving this is imaging. And it's the ability to image an incredible number of single spots of DNA, of single DNA molecules. And we can now sequence, on the order of billions of DNA molecules all at the same time in one image, watching this happen. So you collect the images, there are stacks of images you might extend for 100 bases, and you have 100 packed images of just looking like this, except it's much bigger and it's much denser information, and you can then capture each spot, you can follow it through computationally, and you can, at the end of the day, you have, a billion DNA molecules that you just sequenced overnight of 100 bases. So this is really changing everything we're doing. We can now sequence individual genomes for much less money. We can look at how genomes vary, but we can also do some really amazing study of how the genome is packaged, what we call sometimes the epigenome, or the organization of the genome in an individual cell. So next generation sequencing has really changed how we can do our research. I'm not going to go into this too much, but there are ways to sequence actually DNA methylation, not just get the A, C's, and G's or T's, but you can add a chemical that will change the C's into a different base. And if you do that and you know what the reference genome is, anytime you get to a place where you should have a C, and if you read a T instead, you know that was an unmethylated C. And if you read a C where you expected the C, you know it was methylated. So it's a piece of cake. You can treat the DNA with a chemical that converts any unmethylated C basically to a T, and you sequence it, and all of a sudden you know which sites in the genome had that modification. The other ones are a little complicated, but you can do things like you could take the nucleus and you can permeabilize it, and you can add DNase, which is just an enzyme that will cut accessible DNA. And what that will do is it'll cut any sites that are open, right, where the nucleosomes have moved apart, and it lets an enzyme come in and cut. And when you do that, you can then take all those little bits of DNA and purify them out, and you can sequence them. And you're not sequencing them because you want to understand the sequence of the human genome. You already know the sequence of the human genome. You're just sequencing them so you can count them. You can say how many landed here, how many landed here, how many landed here, and then you know the ones that stack up and you sequence lots of times, those were open in cells. This is my favorite, though. This is something called ChIP-seq. So this is a way to study chromatin structure. The ChIP stands for chromatin immunoprecipitation, and the seq, of course, for sequencing. But Let me see. I think the next slide shows this a little clearer. So again, the idea is we want to learn about the open portions of the genome, the repressed portions of the genome. We want to understand what the functions of all these sequences in the genome. And one way to do it is to look at where are tags. Which parts of the genome have which kinds of tags? And this is critical for anything we're moving on. So if I get through this slide and you don't understand, please ask questions. So you want to know whether this thing has a methyl tag or a phosphoryl phosphate, or you want to know what tag each, and you want to map that across the genome. So how do you do that? You take the cells, and you're actually taking living cells and you're fixing them with formaldehyde. You then break up the chromatin. So I showed you those pictures of chromatin. Actually, you can take that chromatin, you can use either an enzyme that cuts in different places, or you can actually use sonication. You can use like ultrasound to break open the chromatin into little bits. The bits are just a nucleosome with some DNA wrapped around it, okay? And you can imagine you did this with a couple thousands of cells. You've got 10 million cells per nucleus. You have in a tube, you've got trillions of nucleosomes in a tube, okay? Then you take an antibody that recognizes histones, but not any histones. It only recognizes histones that have this particular tag that you're interested in. And you use that antibody to pull out those tagged histones, right? And this is a way to enrich for histones that have this particular tag in the picture is one associated with an active gene or an active promoter. So you just pulled out all the active chromatin, and then you just deep sequence all those little bits of DNA, and we can sequence it billions of times, so you can now count and see where you've got these sequences. You can align those sequences to the reference genome. You already know what the genome is, so every time you sequence a bit of DNA, you can just align it to the genome because you just look for its perfect match. And we're not trying to learn, again, we're not trying to learn about differences in sequences. We're just trying to learn where did you sequence. And you stack up these sequences and you build them into density maps. And these maps basically tell you how many times did you sequence this one piece of the genome. And the pieces of genome that have this particular modification, you're going to sequence again and again and again. Okay? Any questions about that? Yeah.
Real simple. What's deep sequence?
Good.
Is that meaning like using the next-gen sequence?
Yeah.
You're talking about reading down through that sequence that's attached vertically on the next-gen slide.
It's using the next-gen sequencing. Yep.
Okay.
And we sometimes use the term deep sequence because, well, it's something we couldn't do 5 years ago. Right? You couldn't sequence something deeply. You could just sequence a few things off the top and you wouldn't really get much information. But now we can, so a shallow sequence might get 1,000 reads. Now we get hundreds of millions of reads and we get deep data. It's not a very informative term, I realize. So probably, now that we have next-gen sequencing, I should just say sequence the enriched DNA. So here's this picture of cells differentiating that I showed you earlier. So this might be an embryonic stem cell. Pluripotent can form any type of cell in the body. This is hematopoietic stem cells. It still has potential, not as much potential, right? Hematopoietic stem cells can form the blood cells. Here's a B cell that's really fixed. So let's take a look at what does it look like when you map some of these modifications? So the x-axis here is the genome. I'm not going to show you the A's, C's, G's, or T's, but this is probably 100,000 bases along the x-axis. And I've kind of, rather than showing you the letters, I've just shown you, well, I told you we can figure out where the protein coding genes are. Here's the start of a protein coding gene, and here are its exons that code for protein. Here are some things called CPG islands that have lots of CPGs or CPG-rich. The name of the gene is PAX5. PAX5 is a master regulator. You guys have probably heard about this in previous lectures, but maybe not PAX5, but PAX5 it's one of those genes, it codes for a transcription factor that will drive cells into a particular fate. PAX5 happens to drive cells to become B cells. You guys might have heard of MyoD, which was one of the first ones found. It drives cells to be muscle. This one drives cells to turn into B cells. So in embryonic stem cells, if you look at the patterns of these different marks, I'll take you through. So here's the one that's sort of associated with active gene promoters. So the starts of the gene where polymerase comes in, it starts to make RNA. You can see nice peaks. So we sequenced, when we did this, pulled down with an antibody against this active tag, we sequenced it again and again. Here's a repressive tag, though. I use stop and go, green and red. The repressive tag for K27ME3 This is a polycomb repressive modification. I showed you that real compact ball. This is one of the marks associated with that ball, also sitting right over the promoter. This one's associated with genes that are transcribing and making protein, making RNA to make protein. Nothing, right? This one's flat. So this is what the maps. This is an example of a map. This is a characteristic pattern that we found quite a few years ago now, actually. We call it bivalent because at the time we were very surprised because you didn't expect to find both an active tag and a repressive tag at the exact same place. But it turns out that all of the master regulators have this in embryonic stem cells. And we're realizing that this is something that keeps these master regulators poised to come on later. Okay? So I'm going to show you now this same locus. Now remember, the DNA hasn't changed, but the chromatin's changed a little bit, right? Now we're looking at hematopoietic stem cells. The polycomb's spreading out. It's getting a little harder to turn this gene on probably because there's a lot more repressive tags. But there still is a little bit right at the promoter of this activating tag, so it could come on. I don't have it here, but if I showed you a muscle cell, you would see that this green is totally gone and this red is spread all across here. It's totally shut off, okay? But what I'm going to show you instead is a B cell. In a B cell, you totally switch this. Now the promoter, again, is very strong. This tells you that polymerase is transcribing across the DNA. Polymerase sort of leaves this mark as it goes across the gene. And you've lost the repressive mark. So this is now a gene is switched on. So this is the basis of the chromatin mapping and of the epigenome. The DNA stays the same, but in different cell types, it's radically different how it's decorated and organized into chromatin. And this is a pretty simple picture just to kind of give you a sense that you have these master regulators. They've got both go and stop genes. They're poised to come on later. If they become a muscle cell where it's not needed, you lose the activating mark. It switches to this off state. And then if you turn into a neuron, you might need the gene. You switch it to an on, a go state. Okay, but I want to show a little bit now, in the last little bit, about what we can learn sort of beyond the genes. I told you that the genes are 1%. You're wondering what else is out there, right? So when cells differentiate and commit to different fates, I've talked a lot about epigenetics and chromatin structure, and I alluded a little bit to the transcription factors, and I just want to go back to the transcription factors. Cell states are fundamentally encoded by the transcription factors that they have. Fundamentally, what makes an ES cell an ES cell is it has OCT4, SOX2, NANOG, these ES cell transcription factors binding to just the right places in the genome and turning on those genes. Okay? Now, when a cell turns into endoderm or mesoderm or ectoderm, when it differentiates, it turns on different transcription factors and it turns off of the ES transcription factors. And now it is expressing those transcription factors, and those factors are now programming it to turn on mesoderm genes. Okay? And you're like, but why did he tell us so much about chromatin if it's really just the transcription factors? So the transcription factors dictate the state. But what's really interesting about chromatin, I'm going to show you a little more, is that when transcription factor turns on just the right genes in development, chromatin will shut off everything else. So I think that's the key thing of epigenetics. Transcription factors tell you what state you're going to be, and then chromatin will maintain and sustain that state. Epigenetic memory. Okay, so I'm going to take you through that a little bit. So where does a lot of this action go on? It goes on at enhancers, and you guys are probably familiar with sort of fundamental issues of how an enhancer works. It turns out that a lot of genes and transcription factors, they work through enhancers, which are far away from the genes. It's not just the promoter that turns a gene on or off, it's these enhancers. And how the enhancers work, the transcription factor binds to enhancer and it turns on the gene, and it's really complicated because some of these really important genes like sonic hedgehog are dependent on enhancers that are like 1 million bases away. So we have examples of this, we know that's how it works, but how are we supposed to find an enhancer to genes if the enhancer is 1 million bases away? How do you get a sense of where are these enhancers and how many are there? So that's what I want to talk to you now about, how chromatin mapping has been particularly powerful. So here's another one of these maps. X-axis is the genome. Here are a couple genes. They're bivalent in ES cells. When the ES cells differentiate into neural progenitors, they turn on these genes, and all of a sudden, look, you see these other markings, other kinds of tags arising here, right? In the middle of the desert, far away from the genes. Turns out this happens all over the genome. And these tags tend to correlate with the small number of enhancers we already knew about. So it turns out these tags are lighting up enhancers. So not only can we learn about what these tags are doing because we know which enhancers we're expecting, you see known enhancers have these tags, but now that you know the epigenomic structure of enhancers, you can all of a sudden begin to identify across the genome how many enhancers are there. And I'll just kind of cut to the chase, but one of the big reports of ENCODE was that, in fact, the human genome contains something on the order of 1 million enhancers. So I told you there's 20,000 genes. It's kind of disappointing that we're not much more complicated than a roundworm. But the really amazing result of just the last couple of years is that we may have just 20,000 genes, but there are about 1 million enhancers turning those genes on and off in very complex manners. Now those, we can begin to understand which transcription factors are regulating these enhancers, and then who are the genes that these enhancers are regulating, although that's a big challenge. But now we're getting a pretty good sense of at least where are these sites in the genome that look like enhancers. This is just to tell you that enhancers are really tissue specific. Enhancers flicker on and off like light switches in a very tissue specific manner. This is from a study that we published about a year ago where we looked at all these different cell types. And what we found is that each of these little rows, and these are clusters, this is about 100,000 enhancers in different rows here, clustered by their tissue specificity. Most of those enhancers only come on in one or a couple of cell types. They're incredibly tissue specific. They're flickering on and off. We think they're really critical at the basis of these exquisite cell type specific programs and gene expression and how cells look so different from each other. Here's just different enhancers specific to lymphoblasts, B cells, or to liver. So, why do we think this is important why do we think this is real, and why do we think this is important for human disease? And it turns out that there are some really intriguing correspondences between where these enhancers are in the genome, all throughout what we used to think of as junk DNA or dark matter, and where human geneticists are finding sites in the genome that are predicting disease. So I'm going to go through that quickly, but I think this is a really important point. Turns out that there are lots of differences between individuals in this room. Our genomes are not exactly the same between each other. And there are these variants they called single nucleotide polymorphisms. And they've sort of mapped where the variants are across the genome. And then they did these genome-wide association studies where they compared people with diabetes to controls or people with multiple sclerosis to controls. And they tried to identify those variants in the genome that contribute to the risk of or correlate with one disease, right? So what was really striking is that only about 7% of these variants that seem to associate with disease are in genes. Those variants are not sitting in genes. They're sitting in the dark matter. They're sitting far away from genes. But what was really exciting to see in the last year or so is that it turns out these variants that are associated with human traits and disease are enriched in those enhancers that we're finding within the ENCODE and other projects. And so it's quite interesting because if you took a couple traits, so if you took like traits related to blood cell phenotypes, the variants, those base differences tend to occur within enhancers that are active in blood cells. If you took some autoimmune diseases and you asked, where are the variants associated with these diseases enriched? Turns out they're sitting within enhancers that are active in immune cells. So it makes a whole lot of sense that these variants associated with disease-based differences, genetic differences, are sitting within open chromatin and DNA that's sort of open and accessible and seems to be functioning as an enhancer in a related cell type. This is actually a big deal because you take these avenues of research and you sometimes kind of focus on your own science. It was really nice to see validation that the epigenomic work, finding these enhancers and finding all these sites throughout the dark matter way away from the genes that seem to be important. It was really spectacular to see that those sites were starting to line up with the sites that human geneticists, disease medical geneticists, were finding to be important base composition for human disease risk. Yeah.
What's a GWAS?
Genome-wide association study. Yeah. That's how they did, that's how you find these SNPs. Yeah, this was not one of the slides that I prepared specially here. Yes?
I have a question that's kind of bigger, and that is, okay, so all along the way, you're getting stuff switched on and switched off because of what's in the introns, right?
Intergenic regions. They're intron. Yeah, some of them are in introns between the exons, but also outside of the genes.
Well, my question is this: how much of this control is external to the cell and the result of the medium in which the cell is sitting, and how much of it is just automatic? Like, this is the program, you run the program.
Mm-hmm. Mm-hmm. Development is. Yeah, it's an incredible process. It's very early on. Cells respond to their environment and adjacent cells and where they are in a milieu, and there are gradients that are in the embryo that cause cells to differentiate in different ways. So at the end of the day, yeah, cells are pre-programmed a little bit as they start launching down different directions, but the milieu and the environment and who's neighboring and who's touching them has an enormous effect, signaling into the cell and into the nucleus to tell them what they should become. Absolutely. Yeah.
I think I just blanked out for a second. I apologize. Enhancers, are you saying that this is mutations in the enhancers, or are you talking about the epigenetic?
Yeah. So we found these enhancers because they're sites of open chromatin that have characteristic structures that look like they're regulating genes from a distance. The human genetics field didn't know what they were. They just found these same sites because it turned out that a base difference between individuals in this site predisposed you to MS or some other disease. So it was just striking to realize that they're finding the same sites in the genome. We think they regulate genes from a distance. They're seeing that they're important for human disease. You put the picture together, you realize that a lot of, there's simple traits like eye color that's a couple genes. But diabetes and other diseases are not simple. They are dependent on many traits, many different determinants, and it's looking like they're dependent on a lot of switches in enhancers that are subtly regulating gene expression. So I'm kind of going a little bit along on time, I think. So I just want to get back to this. So I told you how these transcription factors can act at enhancers in complex patterns to turn on just the right set of genes. I've told you there's lots of enhancers throughout the 3 billion bases of the genome, and they turn these guys on, and different transcription factors program them. But I also kind of hinted that, but chromatin had this really important epigenetic way of sort of, not in ES cells where you have potential, but here of sort of restricting the potential of cells. So how does chromatin act as cells differentiate and commit to now confine cells to that fate which they've just established? So I showed you one thing, which was at a promoter, a bivalent domain, going from poised to either off or on. But here's some other very recent data that we found. And that is, again, this relates back to this repressive polycomb mark. This is a mark of a classic epigenetic repressor. It's the tag that recruits this protein that compacts and shuts down the chromatin. If you look at ES cells, these pluripotent cells that can form anything, and you ask, where's polycomb sitting? It's tightly confined to promoters where it's sitting in the bivalent domain that I just showed you previously is poising genes. It's not keeping genes shut off, it's poising them. But if you now look at a cell, a differentiated cell, and this pretty much accounts to any differentiated and committed cell you look at, any cell that's its fate has been set. This polycomb mark is now spreading across the genome. So anytime you have a gene here's a gene is on, but everywhere else you have polycomb shutting down. So anywhere there's no gene, anywhere you haven't turned on a gene or you're not utilizing an enhancer, polycomb spreads across and shuts down that region. And you can imagine that it's probably not just spreading, it's probably bringing in the DNA and compacting it like in this picture. So this is, at its heart is epigenetic regulation, right? The transcription factors are telling you which gene needs to come on in this neuron that you've just made. And polycomb is coming in and shutting everything else off. So that neuron is going to stay a neuron throughout your lifetime. And this is just another way of looking at it. It turns out that ES cells, which are pluripotent, have all these all this potential for differentiation, can really turn on any gene they need to turn on depending on their fate, have some active regions that are transcribing genes, but all of the rest of the genome is sort of sitting there in an accessible way. It hasn't been shut down yet. And then after a cell differentiates and commits, you still have these active regions, and you probably have just about the same amount of active region. The nucleus looks similar. There's the same DNA sequence. But what has now happened is polycomb has come in and it's just shut down huge swaths of genome that are not being utilized in that cell.
When you're using the word polycomb, at first, earlier in your lecture, I thought you were talking about so a bunch of nucleosomes were being compacted. Are you using it as a protein now? Or
So polycomb is a whole, actually, if I had more time, I would tell you about how polycomb was discovered as in model organism fly, as a defect that would cause fly body parts to turn into the wrong body part. It would lose its memory of what it should be. A wing would turn into a leg or something. Polycomb is a gene. It also makes a protein. So polycomb was first discovered in flies, but the protein that polycomb encodes, that it makes, is the protein that was added right here to make the chromatin shut down.
So you're calling the protein polycomb as
It regulates Hox genes. It's what sets the memory. When you decide which Hox genes to turn off and on, polycomb shuts the ones that off, and it just keeps them off. It's related to Hox. And when you mess up polycomb, you totally mess up Hox regulation. So you need the TFs to turn on the right Hox, and you need polycomb to keep the rest of them off. And this is structurally sort of a vision of how it happens. Yeah, so I told you about a lot of research. We have a great community. I kind of mentioned my collaborators at the Broad Institute and MIT and MGH. We have a great community here, and we're funded by Howard Hughes, by NHGRI ENCODE, and by the Epigenomics project at the NIH. Yeah.
I just wanted to go back to something you mentioned earlier on with a little bit of that skepticism as you approach the studies of epigenetics, because it's everywhere. It's in the news. As we approach our students, I think it's exciting to learn about what's happening. Do you foresee really the potential use of the science in treatment and therapies if we can control some regulation as we treat most things symptomatically? Will this revolutionize? Do you really think it can revolutionize medicine? Since I think it's sort of promoted, like if we know this, then we look at all of these things we can fix.
Well, let me start small. Let me say there is one area that's clear as day. This is going to have an enormous impact, and I would bet money on it. That's cancer. Turns out that when you get mutations in cancer, there's no drug to fix a mutation. You can't correct the DNA-based mutations. But we're now learning that there are enormous epigenetic defects that are essential for a cancer cell to proliferate. There are now, it's amazing, pharma companies and biotech companies are quite interesting. Once one or two start doing it and everybody does it. So we now see, I've been struck at the number of pharma companies that are now developing drugs that target those histone deacetylases or histone acetylases, or the ones that put the methyl tags on, or this tag. There are enormous programs in pharma. We're getting drugs now. Some of the drugs are approved for cancer therapy, and we have a whole pipeline of new drugs coming through, and I'm incredibly excited about that. But you wanted to ask me more, and that is, can epigenetics allow us to understand the environment and how it's influencing our fate, and whether we're going to get this disease or we're not going to get this disease? I think it has a lot of potential. It's a long haul. It's very hard to make really accurate measurements. It has to do with, if you want to study humans, these GWAS studies, you asked me what it meant, the genome-wide association study, they study human genetics. The sample sizes are enormous. You have to have hundreds and hundreds of individuals, and you need to sequence their DNA or use other ways of detecting which base they have at those sites. And then you have to have hundreds and hundreds of controls. DNA sequence is really accurate, right? I mean, it is digital. You know if there's a base difference, if it's an A here and it's a T here, you know that. It's digital, it's clear as day. Yet you need these enormous samples. Remember the data that I showed you here. I mean, this is, kind of messy, right? I mean, this is the difference between an ES cell and an endothelial cell. So it doesn't get any bigger than this. I mean, this is an enormous difference But can you imagine trying to compare an endothelial cell from me and from you and from 10 other people and trying to look at this data and try to understand who's had some environmental insult? Can you imagine the population sizes to be able to do these studies on a human? I think there's a lot of information there and there's an enormous amount of potential. But the road is really challenging, so I'm sometimes disturbed when I watch Nova or something like that and my colleagues are talking about how we're just on the cusp of understanding why, diabetes. There has to be something there. I mean, we know you guys have probably seen the Dutch famine, the Dutch winter when there's this famine, and you know that 50 years later there's some memory that in utero there was a period of in utero starvation, and the clinical data is clear as day. The epidemiology is clear as day. There is something being remembered. Do I think that this is going to figure that out? I don't know. It's going to be awfully tough. I certainly have colleagues who are trying it in the right way. And the really rigorous colleagues I know actually typically come back and they say there's no difference. We're not detecting a difference, which I think is right to say. If you don't detect a difference, you say we don't detect a difference. There are occasional studies that are finding these tiny, tiny differences, and I haven't seen them replicated. It's not to say that it's not true. It's just going to be a long haul to really definitively identify those differences. The genome is a big place. It's going to be a long haul before we get there. I think there's a lot there, but I just think we have to. The reason why I'm a little skeptical is it's just we have a lot to learn, and this is a hard thing to study. Genetics is going to get a lot of places first. They're not going to get everywhere, and I think the next wave probably will be epigenetics, but it's going to take some time.
Just a mundane question, but it seems counterintuitive to have an enhancer a million bases away from a gene, unless it's all on a spool. Being enough of a fisherman, if I want to tease out the line in one part of the spool, it makes the line tighter someplace else. So is there some sort of correlation between tight winding and being further away allows you to loosen it up again? Does that make sense?
Well, let me say that in a lot of cases, probably what's happening is you're looping around. You're making big loops, and the enhancers are probably coming back to the gene and regulating the gene. A lot of loci have really complex regulation. Like there's a gene like MYC, and MYC needs to be turned on in like 30 different contexts. And it turns out it has different enhancers at different places, and they probably all loop around in different contexts to turn on MYC. And maybe you need a lot of landscape to do that. I don't know. But I agree, it's really counterintuitive that you would put these enhancers so far away. Yeah.
Does the DNA methylation have a different role in differentiation from the histone modification? Or are they sort of overlapping?
That is a question that's of great interest to the field. There are some parallels and some clear distinctions. DNA methylation silences a lot of parts of the genome. It silences some of these repetitive elements, transposons, and these strange elements that invade our genome and have many copies of them. And DNA methylation silences them with particular chromatin pathways. On the other hand, this polycomb that I'm showing you here, these regions really don't seem to be regulated by DNA methylation in normal contexts. So DNA methylation has a role, and it goes to a lot of these repetitive elements, and there's other things it does. And in those cases, it recruits a particular flavor of tag, a particular flavor of the chromatin tag, histone modification tag. But a lot of the other different flavors of histone modification, they don't seem to have, they don't seem to relate to the DNA methylation. I would also say, if you go back in organisms and you look at Drosophila or something like that, you can see that most of these histone modifications I've talked about, you can find them in Drosophila, and they seem to be doing kind of similar things. I told you about polycomb. There's also some of those other tags in Drosophila too. But Drosophila don't have DNA methylation. So it's a little different what it's doing. You know, as your genome gets really big, you might need DNA methylation. It's related but different. That's a good question.
Yeah, I was just thinking this is sort of a two-dimensional model of the genome. Is anyone moving towards a three-dimensional understanding and trying to?
Yeah, that's a complicated problem, but there's some really smart people here at this that are doing. There's actually, you might enjoy, there's a TED talk by Erez Lieberman that's kind of entertaining, because he I don't know how accurate it is, but he has a model of the three-dimensional folding of the genome. He made a bunch of measurements about how far this locus is from that locus, from this locus, and he tried to fold it all into, I think, and it's like a fractal molten globule, and he collaborated with this guy, applied mathematician here, and if you like math, then you'll probably like, and he's a really, actually, you know what? I take back the TED Talk. I realize this guy's kind of a dilettante, and his TED Talk is about the usage of language throughout history. So it's not, he did give a TED Talk, but his TED Talk wasn't on the molten globule genome structure. But yeah, a lot of people are studying that. It's really important. Any other questions?
Thank you.
Oh.